You can tell when it's a good meeting when people really have a lot to say to each other. And this is obviously a really good meeting. And I'm really delighted now to introduce Peter Kim, who's done huge amounts of really interesting work. He tells me he's been 16 years in MIT, and then he was at Merck, and now he's transitioning out and trying to find academic home, presumably beginning with M as well, based on history. Peter, it's all yours.
Well, subsequently, Don Wiley's group also solved the structure of the sprung form, if you will. And indeed, what they found was that this part of the protein did fold as a three-stranded coiled coil. So it provided structural evidence that this hypothesis was correct. And subsequently, Judy White's group at the University of Virginia was able to take advantage of the fact that because there were two crystal structures of each form, she could predict where to put in an amino acid called proline, which disrupts helices. And that protein containing the amino acid substitutions for prolines would still fold and bind the receptors, but it was not function as a fusion protein, presumably because the protein could not form these helices because the proline disrupted the helix. And so that provided functional evidence that the spring-loaded model was correct. Now, at the time that we made this hypothesis and speculation, we also noticed and pointed out that the architecture of many other membrane fusion proteins from other viruses was very similar to that seen in the influenza hemagglutinin. Namely, there was a receptor binding domain, which is shown on the left-hand side, but then the part that I've been talking about, which is the part that's anchored in the viral membrane, was indicated here in purple. The hydrophobic fusion peptide that I've been talking about is indicated here in green. And remarkably, in many of these proteins, immediately adjacent to that hydrophobic fusion peptide was a region that, again, was strongly predicted to be a coiled coil. And so at the time, we speculated, that perhaps many other viral fusion proteins would also use a spring-loaded mechanism to infect cells, including the fusion protein of HIV, which is referred to as the HIV envelope, with the GP120 subunit that binds to receptors and the GP41 subunit, which is responsible for causing the membrane fusion. Well, we went on to study the HIV protein, and this is the work of Steve Blacklow and Min Liu when they were postdocs in the lab. And what they did was to take that HIV protein and to carry out what we call protein dissection, to basically take that protein and subject it to proteases, which are enzymes that degrade proteins. And when they subjected this protein to an excess of proteases that degrade proteins, what remained behind was a very stable core, a core that was resistant to further digestion. And that core consisted of two parts, one from the N-terminal region of the protein and the other from the C-terminal region of the protein, shown here in orange and blue. And without going into the details of how they deduced this, using biophysical studies, they came up with a model that the orange part formed this three-stranded coiled coil, and the blue part folded in an anti-parallel way, that is, going in the opposite direction on the outside of this three-stranded coiled coil to form this so-called trimer of hairpins, so that you actually get a hairpin structure. Subsequently, our lab, Wiley's lab, and Liu's lab solved the X-ray crystal structure of this core. And sure enough, there was a three-stranded coiled coil in the middle, and packed on the outside in an anti-parallel direction were these peptides from the C-terminal region. So the trimer hairpin structure was borne out by crystallography. Remarkably, work from our lab and many other labs went on to solve the crystal structures of many other membrane fusion proteins. And this just shows a compilation of some of them. And in all of these cases, although they used different ways of doing it, the proteins managed to have this trimer of hairpin structure in which the amino terminal end near the fusion peptide forms a three-stranded coiled coil indicated in orange, and then you get a hairpin back where the carboxy terminal end C-peptide folds back on itself. So this all then led, taken together, to a model for how it is that fusion actually occurs. And that is that on the surface of the virus membrane is a protein, a glycoprotein, fusion glycoprotein. That glycoprotein binds to receptors on the cell that it's going to infect. That causes a change in the shape of the protein, which releases the spring. And then, in that spring-loaded manner, the protein now becomes a membrane protein in two different membranes at the same time. It's already anchored in the viral membrane, but now it's inserted into the target membrane of the cell as well. And then, in a subsequent reaction, these two parts snap back together to, to form the trimer of hairpins and bring the cellular membrane and the viral membrane together to cause fusion. Okay? And so that was the model. Now, it turns out that prior to the work that we had done, another group, Carl Wild and his colleagues at Duke University, for completely different reasons, they were actually interested in the immunogenicity of this, in antibody reactions to this protein. They synthesized a bunch of different peptides, short pieces of this protein, and they discovered that one of these peptides actually inhibited HIV infection. And it turned out that that peptide, corresponded to a region from this blue helix. And so we thought that one way in which this might work to inhibit HIV infection was that this peptides derived from the C-terminal region here, this blue part, when added exogenously, could actually bind to the pre-hairpin intermediate, and in what's called a dominant negative manner, prevent now this hairpin from forming. And without going through all the results, I'll just say that Blacklow and Liu went on to show that indeed they could identify from their C peptides that they inhibited, and that when they added the N peptides together, they no longer could inhibit, presumably because they were soaking up the C peptides, consistent with a dominant negative manner. Chan and his work showed that if you destabilize this interaction, you destabilize the structure of the 6 helix, but you also destabilize the intermediate and therefore get a weaker inhibitor. Matthew's group at Duke showed that if you isolate viruses that are resistant to the C peptides, the amino acid changes that occur in the virus actually map to the orange helices, really providing strong evidence that this is how this is going on. And Carol Weiss's group used a technique of tagging a C peptide and using an antibody to pull it down, and she was able to actually pull down the GP41 protein, again consistent with this mechanism. And finally, most recently, Bill DeGrotto and his group used immunogold labels particles hooked up to these C peptides and then electron microscopy to directly visualize these peptides at the interface between the virus and a particle that is mimicking a cell. So really, there's a huge body of evidence that this is the way in which these C peptides inhibit this, and by inference, a strong piece of evidence that the pre-hairpin intermediate does indeed exist. Now, one other piece of evidence that this all makes sense is that when we solve the crystal structure of this 6 helix trimer of hairpins, we notice a deep pocket on the surface of these orange helices. And we speculated that that might be a very good drug target because there was a highly conserved region on the surface of these orange helices, and if we bound a molecule there, you could prevent formation of this trimer of hairpin structure. And indeed, Debbie Eckert, when she was a graduate student in the lab, went on to use a methodology that we had developed earlier called mirror image phage display to identify cyclic D peptides that bound to that pocket, and she solved the co-crystal structure to prove that they actually bound to that pocket. And those pocket binders were actually capable of inhibiting HIV-induced fusion. Okay? So really providing strong evidence that this is how it all works. I should say that this is a highly conserved region, and it's highly conserved in many different viruses. when one thinks about it evolutionarily, the protein has to be able to fold in a native state as well as in the sprung state. And so amino acid changes are going to be hard to accommodate. And since I'm in Boulder, I also have to point out the RNA aspect of this story. And that is that it turns out that the region of the RNA that encodes for this N peptide pocket is actually part of a highly structured RNA element called the Rev-responsive element, which is where a protein called Rev binds the RNA of HIV. And so there's yet a third pressure to not mutate this region and to remain highly conserved. Finally, the peptide that I told you about from the Duke workers was actually taken forward by a biotech company called Trimeris, and it was developed as an injectable drug for the treatment of HIV infection. The drug was approved by the FDA, and ultimately the company was bought by Hoffmann-La Roche. And what this slide shows is one of the studies from the New England Journal of Medicine showing that in patients that are resistant to existing HIV therapies, if you have a control group where you tell the physicians to just do the best they can, versus if you add this peptide, you can actually get improvement in HIV treatment. The drug itself is not a very big drug because there are oral drugs that are available, but it is used still for salvage type treatment. And most importantly for this talk, it shows that in humans, the pre-hairpin intermediate is a valid target that can stop HIV infection. Okay, so that's the background for what I would like to discuss with you. And what I'd like to discuss with you, the problem statement is shown here. As you've heard earlier, HIV in the United States and in wealthy countries is actually a remarkable success story of the pharmaceutical industry, where it is now a chronic disease. We've taken a deadly disease and turned it into a chronic disease. And indeed, in the United States, people that are HIV infected die more from cardiovascular disease than from HIV infection. It's a remarkable triumph. That said, that's true in rich countries, but if you look at the world, this is the United Nations compilation. In the world, it's estimated that there are 34 million people infected with HIV. Since there are about 7 billion people on Earth, that means that 1 out of 200 people on Earth are infected with HIV. And clearly, the ideal way to deal with this enormous health problem in the world would be to come up with a vaccine. And that's turned out to be incredibly difficult. Many people have tried to develop vaccines against the GP120, that protein on the outside surface, and the virus mutates. Coming back to Larry's talk, this virus mutates like crazy, and so it can just get around antibodies that are developed against that, and it's really been completely unsuccessful. Other workers, including Merck, have tried to come up with a completely new vaccine, which is what's called a cell-mediated immunity-based vaccine, where basically you prime the body's immune system, its T cells, to attack cells that are already infected. And that vaccine actually worked in monkeys, but when we tested it in people, it actually did not work, and we stopped our phase 2 study. So it's really a very frustrating problem. The question that we asked many years ago, is would it be possible to figure out how to make antibodies that would bind to the pre-hairpin intermediate and in a manner similar to the peptides that I've been talking about, actually bind to that pre-hairpin intermediate and prevent HIV infection? If you think about it, this pre-hairpin intermediate is a transiently exposed structure. So it's not as though it's going to be sitting around a lot in an HIV-infected person, and so you wouldn't expect an HIV-infected person to make antibodies against this intermediate very easily. And I've also already gone through how it is a highly conserved region, so maybe one could be able to come up with a way of actually stopping infection by immunizing people with the pre-hairpin intermediate. But of course, the pre-hairpin intermediate is transient, so you can't use it to immunize people. It doesn't last long enough. And so what one needs to do is to figure out how to come up with a way to mimic the pre-hairpin intermediate with a stable structure. And through our work, we had actually come up with peptides or proteins that actually mimic the pre-hairpin intermediate. And so one of them is shown over here. It's a peptide which contains 36 residues from this orange helical region, and it's fused to a three-stranded coiled coil, which we call here IZ. It's been very well characterized, and one might expect that it would mimic this structure right here. And similarly, in other studies, we designed a protein which contains 5 of the 6 helices in that trimer of hairpins, and again would be and it's very stably folded, and again would be expected to expose a surface against which, if you could raise antibodies, might actually be expected to inhibit the pre-hairpin intermediate. And now I'll make a very, very long, many year, 15-year story short by saying that in spite of really heroic efforts with different peptide models, different adjuvants in many different animal models, we were not able to elicit an antibody response in animals that neutralized HIV in culture. And we were about to throw in the towel when we decided to just go one step further and ask whether or not, if we used these sorts of molecules to select from a phage display antibody library, antibodies which bound to these peptides, could we find any antibody which actually inhibited HIV infection? And so we collaborated with a company called CAT, Cambridge Antibody Technology, which had libraries that expressed so-called FV regions of antibodies on the surface of phage. And we used these as the bait, if you will, to pull out antibodies that bound to them. And then we asked whether any of them actually inhibited. And sure enough, we were able to identify antibodies which did inhibit HIV infection. One of them we called D5. And this just shows that D5, both as the so-called single-chain FV, but also as a full-length antibody, full-length IgG, inhibits HIV infection. It does so weakly, but it clearly did inhibit HIV infection. And so, just as we were about to throw in the towel, this told us that you can actually get an antibody to get in there and bind it if you could come up with the right way of eliciting an antibody response in people. The X-ray crystal structure of this antibody bound to the three-stranded coiled coil showed that indeed it did bind exactly where we thought it would, and that's the three-stranded coiled coil. This most recent structure from the Merck group shows that you could actually get 3 antibodies binding to one coiled coil. And remarkably, the antibody binds to the hydrophobic pocket that I told you about earlier, where we had the D peptides. And it makes intimate contact with the very highly conserved residues of that pocket as we move forward. So together with, in collaboration with David Montefiore down at Duke, who is really the guru in terms of neutralizing activity, we took this antibody and we screened it against a bunch of different pseudoviruses, they're called, of clinical isolates. So what this is, these are clinical isolates from patients infected with HIV, and the envelope protein has been put into a different virus so we can assay it. And each of these clinical isolates are shown here by their name. And so-called clades, these viruses are grouped into different families called clades. And they're also grouped into what Montefiore calls tiers. Tier 1 are easier to neutralize, Tier 2 are harder to neutralize. And what you see is that this antibody, this monoclonal antibody, is in fact capable of neutralizing a wide variety of antibodies, a wide variety of viruses, demonstrating that there is the breadth of neutralization that we predicted from the conserved nature of this region. Although, to be clear, these are weak binders. These are 100 microgram per mil, which is not a very tight binding for an antibody. So we have weak activity, but it's broad activity with this antibody. Okay, but importantly, it validated, these results all taken together, validated the pre-hairpin intermediate as a potential vaccine target. And so, now armed with this, we set out to go back, even though we spent 15 years with negative results, to go back to animals and see whether or not we could actually get a neutralizing response and a polyclonal response. only we wanted to stack things in our favor. Okay? So first we went back and we've been working hard on trying to design better, what are called immunogens, or better peptides to immunize with. We also stacked things in our favor by using a mutant that we had made, which was actually hypersensitive to this D5 antibody. And then to really make sure we didn't miss the needle in the haystack, we purified IgG so that we could concentrate it and look for even a weak activity. And so these are the different immunogens that we designed. This is the one that I've already told you about. We were concerned that at very low concentrations in the body, these peptides would dissociate. They would come apart, and we would lose the structure. And so one of the things we asked is whether if we covalently bound them together, whether that would help with immunogenicity. And then finally, since this part of the protein is not from HIV, and we knew it was eliciting antibodies, we tried to shield it with what's called polyethylene glycol to prevent antibodies from binding there. And we asked what those three things would look like. This is where the mutation is located. It's located right in a pocket, and we had shown that this mutation actually was more sensitive to inhibition by peptides and by antibodies that bound here. And that results, one of those results is shown here, where both Fusion, the drug that's approved, D5, the antibody I've been talking about, are much more effective at neutralizing this mutant virus, whereas this antibody called B12, which neutralizes the outside part of the protein, GP120, it has nothing to do with this mechanism, there's no difference in neutralization. So we had a more sensitive way for looking. And again, as I said, we purified the antibody so we could concentrate it. And so here are the results that, for the first time, we were able to see activity from antibodies isolated from animals that were immunized with our immunogens, which was a very exciting day, in which each of these symbols represents the antisera from a different animal, in this case guinea pigs, comparing the three-stranded coiled coil to the covalently linked coiled coil to the protected one here, using the more sensitive virus here, V570A. And for those of you that are familiar with antibody concentrations, you can see that we're going up to 3 mg per mil. All right, so we're really looking for any activity at all. And we're seeing activity with a more sensitive virus. With the wild-type virus, which does not contain this substitution, we're also seeing activity, but you can see it's very weak. It's hundreds of micrograms per mil. Okay, but we're seeing activity. And it seems like trimerizing, covalently linking these is helping, and shielding this part of the protein is not making much of a difference. So as I said, this was the first time we had neutralizing activity from an immunized animal that worked in this way, and so that was very exciting. We also then went to Montefiore and asked him to test these antisera in different clinical isolates. And what is shown here is 1 dilution. So this is taking antisera and diluting it. In this case, we're diluting it 85-fold. And when you dilute it 1 part in 85, you can get 50% inhibition of this clinical isolate. So what we're seeing here now is activity in unfractionated antisera from the guinea pigs, which is able to, in some cases, neutralize the virus at some modest dilutions. So armed with that, we've now continued to move on, and I just will close by telling you about our latest unpublished results. And the significant step that we've taken here is we focused on this trimerized, covalently linked coiled coil of 36 residues. But now we've asked whether or not extending that to include more of the pre-hairpin intermediate might actually help. So we've now made this immunogen, which contains 51 residues, and it's linked by a chemical in this case called chems triacids, hence the name KTA. And we've compared this versus this in different animals, and I'll just show you the results for non-human primates, since that's obviously most relevant for humans. And so the experiment that was done here is to take either that trimer or the longer version, 51 residues, and to dose, in this case, rhesus macaque monkeys, and we've primed them with 2 doses here, and then waited a long time. And so you see the last dose was 40 weeks after the initial immunization. And you just heard a talk about adjuvants. This includes a standard aluminum adjuvant that we use at Merck, but also an experimental adjuvant called ISCO matrix, which we found is very effective at eliciting both a T cell and an antibody response. And went into rhesus macaques. And the results are shown here. In this case, on the left-hand side, is neutralization, again, is 1 over dilution, using either the 36 residue covalently linked or the longer 51 residue immunogen. I should say that in the pre-immune sera, that is before we immunize the animals, there's no activity that's observed. And the limit of quantitation, the limit at which we have confidence that we can say something, is shown with a dotted line. And so as you can see, even with a more sensitive virus, we're really right on the edge, okay, of actually believing that we have something real. But we do think this is real because the pre-immune sera don't show it. And here in a clinical isolate, using a very sensitive assay that David uses, again, we're seeing hints of activity in the rhesus macaques. Now, I want to say, this is a long way away from where we need to be, all right? But what's encouraging is that we think that we now have a handle in rhesus macaques, in monkeys, to actually try and optimize what we've seen here. So we have a little signal that we can try to optimize. And so to summarize, the dream really is to try to get antibodies against the pre-hairpin intermediate. And the drug, Fusion, really validates this pre-hairpin intermediate as an effective therapeutic target in humans. The antibody D5 that we isolated and that we showed by crystallography binds to this pre-hairpin structure and also shows broad activity, neutralizing activity, shows that antibodies directed against this can be neutralizing. And now we have finally obtained weak, and I want to emphasize it really is weak, but we believe real neutralizing activity in non-human primates against that pre-hairpin intermediate. And so maybe, and Steve, maybe you can help us with the use of better adjuvants, with different formulations, with the help of the community in terms of thinking about ways of optimizing this response, we might be able to get a more robust immune response. And my own personal feeling is we still have a long way to go, but if we could increase the activity that I showed you in rhesus macaques by about a factor of 100 or so, then I think we would then be in a position to start doing experiments in humans to see whether or not we could get that sort of immunizing antibody, neutralizing antibody response. So I will stop there and just say that this has been a project which has been going on for almost 20 years, and I've tried to reference the publications where it is that they've come. In terms of the more recent work, which has not been published, these are some of the co-workers at Merck and at Duke that have helped with the work that we're doing there. And with that, I will stop and be happy to answer questions. Thank you. Who's got a mic?
Yeah, my question is, so if you do design an antibody or some sort of peptide that targets this region, since it's so transient, are you worried about its effectiveness in finding the target? And have you thought about ways of tethering it to the parts that are not so transient?
Yes, thank you for that question. There were two major concerns that we had when we started out this work. One is the first one that you mentioned, namely, this is transient, and so how on earth are you going to have the time to get in there and stop this process from going on? And the second was, you're talking about two membranes that are coming close together. Maybe you can get a little peptide in there, but how on earth are you going to get a big IgG, a big antibody molecule in there to actually inhibit? And really, I think the best evidence that those are not issues are, first of all, with regard to it being transient, the peptide works in human beings. Okay, so it is there long enough, and it can stop HIV infection in human beings. So we know it's validated in humans. There actually have been some studies that have shown that this intermediate hangs around for minutes, not tens of minutes, but not seconds. So it looks like there is time, but I think the proof is in the human studies. And in terms of the access and the space, that's why it was so encouraging when we actually got an antibody that was full length, that actually inhibited, and then when we solved the crystal structure, we saw there was plenty of room for the antibody to get in there. And so I think both of the concerns, which you raise appropriately, the one of the two that we were very worried about, I think we're in pretty good shape. What we have to do is figure out how to goose the immune system. Right. Yes, that's right. If you could attach something and increase the local concentration, it would be much more effective. From a vaccine point of view, that's not going to help us because we need to elicit the antibody response. But from an inhibition point of view, absolutely. Right. Yes, who has a microphone?
Yeah, my question is, can you put this into the context of the Ian Wilson and Dennis Burton work at Scripps, because they've done a lot of antibody binding to different forms of the envelope protein of HIV, and how does that relate to what you're doing?
Thank you. Yeah, these guys, Dennis Burton, Ian Wilson, Bill Scheif now, these guys have done fantastic work. Many workers in the HIV antibody consortium have done fantastic work where they've been able to pull out from infected HIV patients antibodies and clone the antibody-producing cells, the B cells, and get the sequences and then to solve the X-ray crystal structures of these antibodies and show, in many cases, how it is that they think these are very effective antibodies against HIV. The difference between what they have done, which is beautiful work, and what I'm talking about here, is they have not immunized animals. They've taken antibodies and B cells out of infected patients and identified the antibodies that are very good at neutralizing HIV. Yeah, so I'll come to that. And so the question that they need, now what we need to do as a community is take their work and figure out how to now design agents that will elicit those types of antibodies. So far, that's proven to be not possible. Okay. Now, the question you just asked me, are there antibodies against this region in naturally infected patients? It turns out they're not very prevalent, presumably because this is a transient intermediate and it's not around, but there are some patients where you can identify what looks like antibodies against this region. Yes, thank you. Yeah.
Is there anything in the structure of the A-clade, pre-hairpin complex that would explain why the antibodies don't bind those versus the B and C?
Good question. The sequences are slightly different there, and so it could be that we're going to need to come in with a mixture of an A-clade version as well as a B-C clade version, which would be fine if that's the reason. I don't know that's the reason, but it is true that the sequences show variation there. This region is highly conserved, but within clades there are differences, so presumably the virus has accommodated that. Right. Yeah, Larry's got a question here. Yes, who's got a microphone?
I do. Oh. So I want to ask more about the Merck process that I can imagine but wasn't there, to know why you, so I understand why vaccination is a good idea, but I also understand that having an injectable better fusion that really works is better, it would be good. And when you guys thought of one path or the other, help us understand that process.
Yeah, thank you. Merck has had a long-standing history in HIV research. Immediately following the, I forgot, Chris, you know, after you quoted the Surgeon General, I went back and looked up the Secretary of U.S. Health and Human Services. 1994, Margaret Heckler, then the Secretary of U.S. Department of Health and Human Services, declared in 94, quote, "We hope to have a vaccine ready for testing in about 2 years. Yet another horrible disease is about to yield to patience, persistence, and outright genius." Right? At that time, Merck was actually very actively involved in HIV vaccine research as well as in studying the HIV virus. And there's a seminal paper that was published in PNAS. Irving Siegel was the first author. He tragically died in the Pan Am explosion, in which they showed that if you made a mutant of the HIV protease, the virus was defective. And that led to the HIV protease program at Merck, which ultimately led to development of Crixivan, which was one of the first HIV protease inhibitors. Merck went on to develop Stockryn, which was a reverse transcriptase inhibitor, and most recently, I had the pleasure of watching the final stages of, well, more than the final stages of the development and approval of the first HIV integrase inhibitor, which stopped the enzyme that allows the virally encoded DNA to integrate, to insert into the human DNA by stopping that with a drug called Icentris. At the same time, Merck has always had a long-standing interest in trying to develop a vaccine. And when I joined the company, they had actually already done a lot of the pivotal work showing that you can actually take a replication defective adenovirus, this is for the aficionados, and get it to lead to what's called a cell-mediated immunity response in monkeys, which was a T-cell-based response, CD8 positive, which actually brought down the viral load. And so when you took this, when the monkeys were vaccinated with this, they still got infected, okay, because you're not preventing the infection. But that infection was controlled by the fact that the animal had T cells, which would kill any cells that were infected with HIV. And that's so-called cell-mediated immunity. And I mean, the honest fact of the matter is this company was ready to go forward, and we were actually preparing to build a factory and make phase 3 supplies. I'm not an officer of the company anymore, so I can say with no expectation that we were going to make money. But to do this as really what we viewed as the most important thing to do that we could actually do for this problem. And it was then that we got the phase 2 results, which actually showed that the vaccine, if anything, made things a little bit worse, both in developed countries as well as in Thailand. That experiment, with a slight variation, has just been repeated by the NIH with the same results. The vaccine group actually had more infections than the control group. One hypothesis is that by actually stimulating the T cells that you are trying to stimulate, you're also enhancing the population of cells that are infected by HIV. I mean, the crazy thing about HIV, right, is that you're infecting the very cells of the immune system. And so as you stimulate the immune system, you're also creating an increased number of targets. And so when we got that result, we obviously shut down that program. And we held a summit with thought leaders in the community and basically said that we were prepared to work with anybody that actually came up with a good idea and some good results, but that the problem now is one of basic science. It's not one that industry can really solve. And I still believe that. I think this is a basic science problem. It's also a problem of human immunology. We don't understand. The fact that it works in monkeys and doesn't work in humans means you don't understand enough about human immunology to actually know what you're doing. And so I think there's a lot of basic research that needs to occur still in order to make this happen. If there were a breakthrough, I'm confident that our company would be willing to contribute what we could. Thank you.
For some of these antibodies, they are supposed to be descended from polyreactive precursors. And the idea has been that one of the problems is that these are autoreactive, and what you're dealing with is these are already tolerant to self. So it's very hard to get the B cells that you want to activate off the ground because they're already trying to stop themselves getting off the ground. Have you thought about that, and do you think that's a problem in this case?
Thank you, Pippa. That's a really important question, and I think it's an important area of research as we move forward. There was a beautiful study published in Science recently by Bill Schief and his coworkers looking at the heritage of a antibody that actually does neutralize HIV, starting with the germline and following the progression through. And the astounding result was that the final immunogen, or the final virus, actually bound very, very poorly to the germline starting antibody. And he put forth the proposal, which I think is actually one that's well worth thinking about here as well, that it could be that you need to prime with an immunogen which is different than the final immunogen that you're trying to get the antibodies to, so that you can drive the process along the lines that you want. And so to your point, if there were, and I don't know that there are, but if there were, tolerized versions of that we're dealing with that we're trying to overcome, I think the question now becomes then how can you stimulate that part to now start the progression to come in later with this? And I think those are the sort of things that we may need to do to actually finally crack this problem.
Peter, thank you so much. I mean, that was really exquisite science, real perseverance.